Hello friends, hello students. Today I am going to discuss about molecular techniques that is used for characterization of proteins. So there are so many different techniques that can be utilized for characterization of the protein. Characterization means to find out the structure, to find out the amino acid sequence, to find out the function, to find out the molecular weight of the protein. So the first technique, that is 2D gel electrophoresis. Second technique, that is SDS-PAGE, and the third technique, that is peptide mapping or protein fingerprinting. The next technique is protein sequencing. So there are two methods of protein sequencing. One is Sanger's method and other is Edman degradation methods. So through two methods we can sequence the any unknown protein samples. And the last, that is the mass spectrometry. It is the highly sophisticated technique through which we can find out, identify a protein even at picomole quantity. So first I want to discuss the technique, that is the SDS-PAGE. The word SDS-PAGE means sodium dodecyl sulfate polyacrylamide gel electrophoresis. You know, this SDS, it is act as a detergent. There is a PAGE. PAGE, it is a variant of electrophoresis. It is going to separate the protein as well as the DNA on the basis of size, on the basis of mass. So that mass by charge ratio. So that is with the help of this polyacrylamide gel we can going to separate the protein after putting the protein sample, inside that the gel. So if you see, this is the complete apparatus of SDS-PAGE gel electrophoresis, in which there are two ends of this electrophoresis apparatus. One is cathode and one is anode. So generally this SDS-PAGE, it will provide the negative charge on each type of protein, whether it is a positive, whether it is the negative, or whether it is the neutral. So it gives the negative charge, due to which the protein molecules, it will move from cathode, that is indicated in black color, to anode. So there is movement from top end to bottom end in case of polyacrylamide gel electrophoresis. So if you want to study the structure of the polyacrylamide gel, then we see the movement of the protein. It will start from cathode, from negative terminal, to anode, that is the positive terminal. So when we apply the electric field, in presence of electric field and the buffer, it will move toward the anode. When 70 to 80% area is run by this protein along with tracking dye, then we stop the polyacrylamide gel electrophoresis process, and then after staining with Comasi brilliant blue, then we can observe the protein band. It is given in this diagram, that is blue in color. So we try to find out with the molecular size with the reference standard. Then we can also use the software to find out the size of the protein, that is the size in kilodalton. So this is the whole story about SDS-PAGE, through which we will try to find the size of the protein, the molecular weight of the protein of any unknown protein sample with a reference sample in the gel, that is polyacrylamide gel. The next technique, that is the two-dimensional gel electrophoresis. So you see, I have already discussed the SDS-PAGE gel. Here there is a marker, that is a reference one in the left side of the gel, and on the right side there are different samples of the different proteins. So that marker, that is the ladder through which we are going to find out the size of protein that is present in different samples, that is present in different samples. So you see the B2, B1, like AX1, these are the different samples. Through marker, that is the M, where the different size it is given in the gel, that is 97.4 kilodalton, 66.2 kilodalton, 43 kilodalton and 31 kilodalton. Means the lower molecular weight, the lower size of the protein will move fast, and higher size of the protein moves slow from the well of the protein gel. Then when it completed the 80% area, after that we stain with Pomasi brilliant blue. This is the protein band that is stained with Pomasi brilliant blue. So through this dye we can detect the protein band. If there is any unique band, then we will try to find whether that protein is unique among all the different samples or not. Then what we proceed? We proceed for 2D gel electrophoresis. Why we proceed for 2D gel electrophoresis? Because we cannot predict whether the protein have same pi or have same size. If it may be possibility that protein that have single band, there is a mixture of two different protein have the different pi. But we can say with the help of 2D gel electrophoresis, we will try to isolate the protein, purify the protein, and then we separate the protein in one dimension on the basis of isoelectrophoresis. You know isoelectrophoresis, that is totally based on pi. What do you mean by pi? You know, the pH of the protein at which net charge of the protein is 0. So each protein have their characteristic. They must have a specific pi. On that basis they are known. So when the protein attain a particular pi, then protein will become astatic. So we put the pH strip, that is from 0 to 14, in the isoelectrophoresis machine. So you see in the diagram of the given slide, this is isoelectrophoresis machine. On the top corners, that is this is the isoelectrophoresis machine. So with the help of this isoelectrophoresis machine, we will try to find the different protein have the different pi. So different protein have different pi. So we put the pH strip. This is the pH strip where we load the protein samples. If there is a mixture of different protein sample and have different pi, so we load the sample from that end, means where there is a 0 pH, and then it start to move slowly and slowly in the presence of electric field, and they become static at particular points. Suppose it is a 9, means that protein have 9 pi. It does not means that only one protein is there. There may be mixture of one or more than one protein. So if there is 7, there may be possibility that more than one protein that have different molecular size are arranged there on a particular pi or particular pH. So we can not conclude that there is only one type of protein. There may be mixture of different size of protein, but all protein have same pi. So with the help of this isoelectrophoresing machine, through on the basis of pi, we are going to separate the protein band through protein mixture through this pH strip in presence of electric field. So this is the separation of protein in one dimension on the basis of isoelectrophoresing. Now if there is a mixture of protein at the same pi, then we further separate the protein through the process SDS-PAGE in second dimension. In the second dimension through SDS-PAGE, the protein going to move from cathode into anode end. Cathode is negative and anode is positive at that end. So the protein will move from this point to this point, toward this point, and the protein that have a smaller size, it will move faster. It will move faster. But the protein that have larger size, it will move slower in this gel. Then we get the different band of the protein in polyacrylamide gel. So this is, we can say this is anode and this is cathode. The anode is joined with the wire with that point, and cathode is joined with the point only that point. So it will move from top to bottom. That is the for the final separation of the protein according to size, they are going to separate. So a smaller size, a small fragmented polypeptide chain or protein will move faster. It will on the top toward the anode end, and the larger size protein near the anode end, cathode end. So that cathode that is the black in color, that is the cathode end, and red in color, that is the anode end. But it is joined at that point. But if you see the cathode end, it is joined with the wire with this point only. Now there are two glass that is jointed, we can fix with the fixing agent, and then we put the sample in this well. So in each well we can put the different sample of the different protein. So protein mixture is going to separate on the basis of size, on the basis of molecular weight. So it will move slowly and slowly. What do you mean by loading of the protein sample? We take a mixture of dye, tracking dye, along with protein from the solution and put in the well of this polyacrylamide gel. When we apply the electric current, the cathode end joined to the cathode unit, that is the negative terminal, and anode is joined to the anode end, that is the positive terminal. When we apply the electric current, it will start separating. After completion of electrophoresis, we proceed for that staining process with the help of the dye, that is Comasi brilliant blue. So that will stain only the protein, not the polyacrylamide gel. So this is the story of SDS-PAGE, through which we are going to separate the protein that have the same pi but may have different molecular size, through which we are going to separate the protein in second dimension. So this is the combination of the the 2D gel electrophoresis is the combination of isoelectrophoresing and SDS-PAGE. In one dimension, we are going to separate the protein through isoelectrophoresing on the basis of pi. In second dimension, we are going to separate the protein on the basis of size through SDS page. So this is the one of the important technique, that is 2D gel electrophoresis. Now I am going to discuss the next technique, that is protein sequencing. What are different method of protein sequencing? Sanger, he has got Nobel Prize in the field of molecular biology and biotechnology, means he has got Nobel Prize for DNA sequencing, but he has also done protein sequencing. Then there is one reagent that was used by Sanger, that is 1,4,2,4-dinitrobenzene, FDNB. And the second method, that is the Edman degradation methods. So these are the most prominent technique for sequencing of the protein. Now this is Sanger's method. If you see, there is a reagent, that is FDNB, 1,4,2,4-dinitrobenzene. So this is a reagent, that is one of the important reagent for Sanger's method, through which we are going to separate the amino acid level one by one from amino terminal. There are two ends in polypeptide chain of any protein. One is amino terminal end and one is carboxy terminal end. So that will react, this reagent, that is FDNB, 1,4,2,4-dinitrobenzene, that reagent, that is also known as Sanger's reagent, that will react toward the amino terminal end, and one by one it is going to separate the amino acid along with that FDNB, and that cycle will proceed. After each cycle they are going to fragment it, and each and every amino acid is going to find out what is the amino acid sequence. On that basis we find out the amino acid sequence of any protein, any unknown protein. Second is the Edman degradation method. So in this case there is other reagent, that is phenyl isothiocyanate. This is the reagent that will react with the amino terminal end of the polypeptide chain, and one by one it will degrade the it will separate the amino acid from amino terminal end, and then one by one also we get the amino acid. What type of, which amino acid is going to separate, we will able to know the particular amino acid in the same way, just like Sanger's methods. But reagent is different, that is phenyl isothiocyanate. So that phenyl isothiocyanate react and produce the product phenyl isothiocyanate derivatives along with the amino acid. So this is the most important technique for protein sequencing. Now this is the two methods I have already discussed, Sanger's method and Edman degradation method. Next method for the characterization of protein, that is next method for characterization of protein, that is protein fingerprinting. Protein fingerprinting is also known as peptide mapping. I have already discussed the 2D gel electrophoresis. It was given by O'Farrell's, but protein fingerprinting, it was given by Ingram. So protein fingerprinting, generally if we explain with the suitable example, suppose we are taking the blood sample of two different person, and then we isolate the RBC of two different person, one is sickle cell person and one is normal person. Then after taking the isolation of the RBC from the blood, we isolate the hemoglobin. One is sickle cell hemoglobin and one is normal hemoglobin. Now we will cleave the polypeptide chain of hemoglobin. Then how can you cleave? There is one enzyme that is known as trypsin. So you must know trypsin always cut with where there is a lysine or arginine that is present on the position in polypeptide chain. Where there is lysine and where there is a arginine, only that position trypsin will cleave the peptide bond, and we get the different fragment of polypeptide chain of the hemoglobin, that is one is normal hemoglobin, one is sickle cell hemoglobin. Then we proceed for separation purpose. Then the same way we proceed paper electrophoresis. In presence of electric field, we quickly separate the polypeptide chain through paper electrophoresis, or we can also use paper chromatography. So this paper chromatography, that is best utilized for separation of fragmented polypeptide of the hemoglobin. One is normal hemoglobin and one is sickle cell hemoglobin. Then after separation on the paper chromatogram, we will use a dye. You must know what dye we will use. It is a ninhydrin. When we put a spray of ninhydrin on each paper chromatogram, then we find the spot of the protein. Then we will try to match the each spot, whether that spot are similar. Then again we match the other spot, it is similar, or it is similar or not. If it is dissimilar, means suppose this is the dissimilar one, then we will focus on that protein spot, and then we take out the protein from this paper chromatogram, and then we purify that protein, and then we put for again iso electrophoresing and SDS-PAGE, that is 2D gel electrophoresis. And if finally, if again we want to purify and separate the different fragment, then for purification we again HPLC technique we can use for final purification. Then we put the sample inside the mass spectrometer, that is MALDI-TOF. It is a technique through which we will find out the protein amino acid level with the reference compound of the protein, through which we can match the amino acid sequence level. What is the percentage of matching? On that basis we will try to predict the structure, the function, the helical pattern, the alpha helix or beta pegylated structure are there in that unknown protein samples. So finally, with the help of MALDI-TOF, matrix assisted laser desorption ionization time of flight, matrix assisted laser desorption ionization time of flight, through which we are going to finally separate the protein at amino acid level. So this is the protein fingerprinting or peptide mapping. This is the protein fingerprinting. Next technique, that one is the last one, through which we will find the protein at whether present in the any compound. So I have already discussed these things that we will again proceed the specific spot. If there is a specific appearance in the spot, then we take out from the gel, and then we isolate and purify the protein from the gel or from paper chromatogram. Okay. After isolating the protein from paper chromatogram, we will proceed for 2D gel electrophoresis. Then again we refine the protein, whether there is a different one or same. If there is a different mixture, again we compare with the two different sample. One is sickle cell and one is the normal person. Then if it is the different one, the protein, then we take out the protein from the 2D gel electrophoresis gel, that is SDS gel. Then we put that protein sample in mass spectrometer. That mass spectrometer already I have discussed MALDI-TOF. With the help of this MALDI-TOF, with the help of reference sample and software for the comparing with that unknown protein sample, we will able to know the amino acid pattern, whether hydrophobic amino acid is there or hydrophilic amino acid, where the basic amino acid is there in polypeptide chain, unknown polypeptide chain, or acidic amino acid is there. So try to find whether there is a presence of aromatic amino acid. On that reference sample will match the percentage matching of the unknown protein amino acid, and then we find out the helical structure as well as molecular weight of the protein. So with the help of software through in silico prediction, we will try to find the function and structure of the protein. So if the 90% or 99% similarity, then we can say it is the particular function of that protein. Next, that is the last topic for characterization of protein, that is mass spectrometry. The mass spectrometry, it was given by J.J. Thomson for identification of the protein, for characterization of protein, for quantification of the protein. So this mass spectrometry, it is a most powerful technique through which we identify the protein, whether the protein is present or not. Then we quantify the protein, means amount at picomole level. What is the basis? First, we ionize the sample. How can we ionize? With the help of mass Malditov or through electron spray. If you see the different component that is of the mass spectrometer, the first component, that is source of ionization, and second component, that is the analyzer unit, and third component, that is detector unit. So if you divide the different component of mass spectrometer, then the first step, the sample should be prepared and should be ionized from condensed liquid state to a vaporized ionic state. So in the presence of laser beam, we can ionize the sample. After ionizing the sample, it will put or inject inside this mass spectrometer. It will attain the momentum. Each and every ionized protein molecule attain the momentum, and in presence of a strong electric and magnetic field, due to presence of a strong electromagnet that is present in the middle of the mass spectrometer, in the vacuum tube of this tubular structure of mass spectrometer, that pass, and there is a differential separation on the basis of mass by charge ratio. So each protein have certain mass. So different protein have the different mass and different energy. The different due to presence of different energy and different mass, when the protein are in charge state, that is in ionic state, either it attain the positive charge or attain the negative charge in presence of laser beam and in presence of high heat. So it will move differentially due to gaining of momentum. So that differentiation, there is a deflection in the vacuum tube near the electromagnetic region. So that deflection can be characterized in the form of mass spectrum. That will characterize in the form of mass spectrum. So this is the first part unit where there is ionization. Ionization is an essential process for detection of any unknown sample or unknown sample protein. So what will happen in ionization? In presence of laser and in presence of high heat, the sample, that is liquid condensed form, is going to vaporize, going to convert into vaporized gaseous form that contain the charge ion. So it becomes charged, and after attaining the charge, when it will move inside this region where there is a strong electromagnet, it will deflect due to differential molecular weight or size as well as energy. So on the basis of deflection, we get the different pattern of mass spectra, and each mass spectra that can be detected by detection unit, and that signals can be amplified with the amplifier. So you know, if you see a chart, then we will try to know with the help of this micro channel plates we can detect we can amplify the signals, or we can use electron multiplier. Now I want to show you this is the complete structure of mass spectrometer, where it is the functional part where there is detection and amplification of the signals, that is the signals of mass spectra. I have already discussed the ionization unit. I have already discussed that is the ionization unit, and then after ionization it will enter inside this vacuum tube, and that will attain the momentum due to different molecular weight and different the energy. It will move differentially in presence of a strong electromagnetic field, due to presence of a strong electromagnet, and then the detection unit will detect the mass spectra. On the basis of mass by charge ratio, it will separate the protein or separate the any biomolecules. So detection unit, the signals it will gain by the amplifier, because it will amplify the signals, that is in the form of mass spectra, and the software will analyze the mass spectra and determine the molecular weight. Not only the molecular weight, the structure of any biomolecules, and the sequence, what are the different components of that biomolecules. So this is the whole story of mass spectrometry or mass spectra. So this is complete process for the characterization of protein. Thank you and have a nice day.